I will take this opportunity to thank all of our visitors for attending this month's version of the ReproNim webinar series. We are delighted to have David Venison and Matt Glasser join us from the Human Connectome project. A bit of news in case we haven't mentioned it since we've last spoken to this attendance group. ReproNim has been refunded for its second 5-year time. So as much as I like to think yay for us, it's really thanks to you, the community, our collaborator projects, our users, and all of our trainees and folks who attend these types of materials that make our work possible and make it possible to be shown in good light to our reviewers. So thank you attendees, and thank you staff, and thank you everyone. So today's presentation is on reproducibility and human neuroimaging, lessons from the Human Connectome project. Matt Glasser is an MD and a PhD in the departments of radiology and neuroscience at Washington University Medical School. He's answerer and explainer extraordinaire on the HCP email lists. Anyone who's joined those lists, certainly hears a lot from him. His interests include neuroanatomy, connectomics, neuroimaging, neuroradiology, medical image analysis, and all of the good stuff. David Venison would take our whole hour to actually fully introduce him. I'll just mention that his work specializes in neurobiology and studies of structure, function, development, connectivity, and the evolution of the cerebral cortex in humans and non-human primates. He's an alumni endowed professor of neuroscience at WashU School of Medicine in St. Louis. I have known David for a very long time, and I'm not really going to spend any time saying much about an anecdote, but it does remind me to be able to at least invoke the memory of Vern Cavanis, who passed away earlier this year, one of the neuroscientists, neurologists, extraordinaire, sort of the old school. And I certainly remember one of David Venison's visits to our lab back in the old days of MGH, probably in the 1990s, sitting around in a restaurant, probably in the north end of Boston, and having a grand discussion about plate tectonics and the cortical architecture of sulcal and gyral development in the human cortex. And these are just great memories, and it's a great opportunity for me to remember to, also invoke, the names of Vern Cavanis and meetings, that we had back in the old days. So without further ado, I will turn the floor over to David and Matt.
Okay, thank you for passing the virtual floor and baton, and I'm delighted to talk about reproducibility in relation to what we've learned from the Human Connectome project. So let's dive right in. See if I can get my slide advanced to actually work. There we go. So the biological finding that David Kennedy asked us to lay out is that MRI-based human cortical parcellations are reproducible. And in part one, I'm going to introduce the HCP-style neuroimaging method and show the sharp contrast it has with traditional human neuroimaging, and I'll illustrate what benefits it does provide, and the spoiler alert that there are major benefits in the context of your ReproNim project. Matt will talk more specifically about our multimodal HCP parcellation and also give examples of other situations in which HCP-style neuroimaging has addressed the reproducibility issue. So the Human Connectome project, for those who are not familiar with it, started more than a decade ago. It lasted for 6 years, and it achieved its objective of acquiring data on brain structure, function, and connectivity in healthy adult twins and their siblings. We got improved scanners at the outset and improved pulse sequences that let us get excellent quality multimodal imaging, 4 hours per subject, which is an unprecedented high for such a large number of subjects, which was 1,100 scanned at 3T, 200 scanned at 7T, 100 with MEG. I won't have time to talk about those. And also extensive characterization in relation to behavior, demography, and genotyping. We analyzed the data using improved pre-processing pipelines, customized for the task at hand, but now general to the community. We achieved better alignment across individuals and to atlases, and better visualization with our Connectome Workbench software. We carried out advanced analyses, including the parcellation that Matt will tell you about. And we shared the data and the methods and tools, and more than 18,000 investigators have accepted HCP and been using HCP data. And there are more than 1,500 publications that now cite HCP data use. And coupled with this process, there emerged from the set of incremental improvements on this or that aspect of processing, what we now call the HCP style neuroimaging paradigm. And it has 7 core tenets that I'll run through briefly, but I'll be illustrating them in a substantive way in the slides to come. First is get as much multimodal imaging data as is feasible given the other constraints of any particular project, and get the data at the maximum resolution in space and time, and that includes using multiband Fmri and diffusion imaging when possible. It's really critical to minimize distortion and blurring of each subject's data, and to respect the geometry of brain structures with what we call the SIFTI gray ordinates that I'll say more about in detail. It's critical to align the data precisely across individuals and across studies, to analyze the results using an accurate brain parcellation, and finally, but not least, to freely share the data, including publication-related data. So there is a reproducibility problem in human neuroimaging, and its roots go actually quite deep. So in the 1980s, Mark Rakel, Peter Fox, and others were requiring PET imaging data that gave what you can see in the upper left, blurry images of brain activity by one measure or another. And by comparing, for instance, visual simulation to fixation only, the difference imaging on the top right is interesting, fascinating at the time, but it's basically noisy and blurry data. And when Rakel and others tried to align the data across subjects, they found that the best they could do in that opening phase was to essentially match the data to a human brain atlas that was published as a book image, and they basically treated it as a shoebox and fit each brain inside to tightly fit that shoebox. And so basically a low-level affine transformation. When MRA came in on the scene in the 1990s, it revealed structure as well as function that was hugely important, and it was possible to get structural images of individual subjects like you see on the upper left. And there emerged a tradition of registering individual subject volumes to a group average volume. And the most widely used one to this day is the MNI 152 atlas. It was initially very blurry, but nonlinear registration algorithm became increasingly sophisticated, and now it is common to use the FNIRT nonlinear registration and register to a group average that has the same size and shape as MNI 152, but it shows a lot more detail. But it does not consistently and cleanly respect the cortical folds, and the failure of volume imaging to respect cortical surface topology is at the root of the problem, but also the solution space. To continue for a couple moments with the traditional paradigm, when investigators got fMRI data mapped onto volumes, they realized that the data were noisy, and even though structural images could be acquired at 1 millimeter-ish resolution, it required large voxels, commonly 4 millimeters in the early days, to acquire workable fMRI data. So in the traditional pathway, volume-based registration to standard MNI brain space was done, and additional smoothing in the volume was done to reduce misalignments, but that led to further blurring. But on that kind of data set, the tradition has been to carry out task fMRI or resting state or structural imaging analyses to generate statistical maps of something of interest, a task contrast, for example, and to show what voxels had a statistically significant effect, and to identify clusters of voxels, and often to summarize them by a single stereotaxic coordinate at the peak of localization, which further loses critical spatial information. Now, interestingly, in parallel with this, starting in the mid-90s, surface modeling came into play. Now, I've been in the cortical surface modeling business literally for 4.5 decades, starting when I used pencil and tracing paper to make cortical flat maps of a monkey. But the critical next advance came with the advent of MRI-based computer surface models, spearheaded by Anders Dale and colleagues. And Marty Serino worked with them to get the first retinotopic maps of visual cortex in humans that were really transformative, because not only were they informative about functional organization, but they illustrated that surface-based analyses provided major advantages. And in the ensuing decades, it is quite common for studies of visual cortex to use surface-based analyses and to demonstrate how it helps in parcellation and functional analyses. The striking parallel state of affairs is that traditional volume-based analysis remains the dominant mode for studies of non-visual cortex, basically the rest of outside the occipital lobe, pretty much to this day. And that obliges us to continue pushing hard for illustrating and documenting the advantages of surface-based analysis. One part of it is that one needs to look at cortical thickness to say what's the spatial resolution that's needed. And on the right, you see a histogram showing that human cortex varies from 1.6 to 4 millimeters, but most of it's 2 to 3 millimeters. But in some regions, the cortex is consistently thicker than average. In other regions, like across the central sulcus, it's thinner than average. But this shows you that conventional MRI, which goes from 2.5 to 4 millimeter voxels, leads to blurring across compartmental boundaries. For the HCP, we were able to reduce the voxel size to 2 millimeters, and even for 1.6 for 7T scans. What is the price that's paid quantitatively? Well, here's a measure of the magnitude of leakage across cortical folds. In the lower right, it shows very little leakage for 2 millimeter scans. It gets progressively worse at 2.5 and 3, but not too bad for those. The real penalty is paid when you go up to 4 millimeters and you see how much leakage there is that causes unavoidable blurring across cortical folds and makes it impossible to tell if an activation is on one side or another of a fold, even though they can sometimes be very different in functional organization. One more point in passing is that even if one has low resolution data at the start, doing the pre-processing using HCP style approaches is advantageous. For example, echoplanar images for fMRI diffusion and perfusion all have stereotyped distortion patterns, much caused by error in sinuses or the mastoids, and the bright red regions in the right images show those distortions on a group average map. But the good news is field maps, if are properly acquired, allow those corrections to be made and compensate for the distortion to a good first order. So in this next slide, I'm going to show a movie that Matt made that really addresses several key issues relating to surface-based registration. So I'm going to talk you through the movie, but basically to give you a heads up, we're going to go from capturing surface representations of an individual subject, inflate it to a sphere, register to a group average, then go back in the reverse direction to a different individual and show how we can carry information from one individual to a group average to another individual. So here you see the white, the green and blue represent the white and peel surfaces, and you capture the full white and peel surfaces. We then average them to get a mid-thickness model that is better for many types of analysis. We focus on the left hemisphere of that mid-thickness, and then inflated, map it out to a pure spherical shape, register on the sphere, which is the best geometry for doing this, register to a group average, and show the overlay between the individual and the very fuzzy group average, which then we bring back to the group average mid-thickness surface, which shows the central sulcus prominently, for example, but the frontal lobe is very fuzzed out because of the high degree of folding variability. We now inflate back out to a sphere for the original individual. We warp to another individual and collapse back into the mid-thickness shape of the second individual. And that illustrates how we can carry topology preserved information from one individual to another. If we had done just volume-based registration for that individual, the overlaid green and lime patterns show the major disagreement that is achieved by volume registration. I'm going to drive this point home in another way, showing here a group average architectonic map of individual postmortem brains of a particular somatosensory area 2. You see it in the upper middle panel. The volume average is spread across many sulci. And if one maps from the volume average to the individual, to the group atlas, one gets in panels D and E a very smeared out representation. Whereas if one does, as Bruce Fischel did, take the same architectonic data, map it to the individual surfaces, one gets a much cleaner group average map. Doing this not only gives prettier pictures, but it gives quantitatively better information. Just very briefly, if one takes volume registered Fmri data and does a task analysis and looks at the Z statistic maps, basically the left column shows the baseline quality of 2 millimeter volume data. If one smooths in the volume, one gets a 20% improvement in the statistical maps. If one registers to the surface and without doing any smoothing, one does even better in the third column. And in the fourth column, doing surface smoothing enhances the signal quality even better. So this is not a novel finding, but it's there have been many studies basically showing that quantitatively one gets more bang for the buck using surface-based analyses. In doing so, one of the challenges we faced for the HCP project was that the surface models were one file type and volume data were a completely different nifty volume type. And to integrate them in a focused way for analyses that are looking only at brain gray matter, we introduced this concept of SIFTI gray ordinates that has vertex representations of each hemisphere and volume representation, but only the voxels that are in the subcortical volumes, not the entire 3D array. And that allows for efficient strategies for intersubject alignment of both the cortex and the subcortical regions. It allows smoothing to be done in gray ordinate space that includes subcortical parcel respecting smoothing within subcortical volumes. Despite these advantages, it was a major issue that we realized early on was that even with surface based alignment using the folding patterns as a guide, one got precise alignment only for selected visual areas like V1, but other areas like MT were smeared substantially across the surface. That's basically because folding variability occurs over most of the cortex. And in addition, the distinct visual areas, such as this one with the red arrows, area 55B that Matt will tell you more about, is variable in its location relative to nearby folds. And so that requires an improved way of aligning across subjects. We take MT as an example, and as already illustrated, a traditional volume-based registration gets poor alignment. Surface-based, using only folding, gets better alignment, but the best alignment and far crisper, as you can see on the far right, is what we call aerial surface-based or aerial feature-based registration. And for the Human Connectome project, Matt combined functional information from resting state networks, Myelin Maps, and visuotopic maps to generate what we call the MSM-all feature-based registration. Those files, when you look in HCP data, have MSM-all in their file names, and the registration pipelines for doing this are given in our Git repository. I'm going to fly by this because we're running a bit late, but basically one gets substantial improvement quantitatively with aerial feature-based registration, particularly in the far right columns for signal quality and distortion reduction. Another very important issue is that, as I said, Fmri data are not only noisy, but there are multiple distinct types. So if you take the basically unprocessed functional connectivity data and correlate one seed with all others, one sees a very noisy, non-neurobiologically appealing pattern. If one removes spatially specific artifacts, the situation gets substantially better. Even better if one uses temporal ICA to remove subject respiratory physiology, and still much better when one removes random measurement noise, and then one gets a neurobiologically very informative pattern. This kind of denoising is various kinds are used in modern neuroimaging, and basically we would say is not all methods are equal, and we encourage a hard look and a careful look at what you use, and very likely we think people will move more towards these spatial and temporal ICA methods that the HCP and successive studies have brought us to. Lastly, I'm going to go quickly through a set of slides that show reproducibility of different measures for 210 subjects that were grouped into a parcellation group that Matt used for class aerial identification that you'll see momentarily, and a validation set that also had 210 subjects. And we're going to compare very quickly different kinds of architecture, function, task, Fmri, and connectivity, and use Pearson correlations between the maps to quantify the performance. So basically, if one takes the parcellation group and looks at the group average folding for the left and right hemispheres and different and compares in the upper and lower rows for the parcellation versus validation group, the correlation is 0.996, very impressively good. For this sulcal depth maps, it's almost as good. And that's basically because, as I've already noted, there's high folding variability, but the patterns of variability are similar across large numbers of groups. When one uses Myelin Maps, one sees that the upper and lower colorful rows get a high correlation of the Myelin Maps, and also high correlation of the thickness maps. So these are architectural measures, and they reveal fine detail. For example, in the somatomotor strip, there's a little arrow pointing to a groove that is actually functionally significant, a measure of the boundary between the upper limb and the face patch in somatomotor cortex. For task fMRI, if we take beta maps for the 210 parcellation group and look at the validation groups, the patterns are very similar, whether one looks at the most reproducible contrast, the median, or the least reproducible contrast. Parcellation and validation groups are very similar, and they reveal a consistent demonstration of even tiny little patches like the white ellipse shown in the right panels. Task fMRI is actually very similar, and if one quantifies the reproducibility, by a different approach, one gets excellent alignment. I think this actually is a repeat slide. Okay, here's resting state. Resting state functional connectivity is also very reproducible, and one can quantify that as well. So basically, we whizzed through a lot of material, but I'll now hand it over to Matt to bring this into the context of multimodal parcellation.
Okay, so the basic point that David was showing at the end there is these different measures that we're going to use to make the HCP's multimodal parcellation are really highly reproducible across these two groups, the 210P and 210V groups of subjects. So it sets a high bar for our expectations of the reproducibility that we're going to get out of parcellations. So to take a step back, though, what do we want in a cortical parcellation? Ideally, it would have the following four qualities: be based on hundreds of precisely aligned subjects, as David was telling us about, and represent the typical areal arrangement of the study population, reflect complementary and converging evidence from multiple modalities across the whole cerebral cortex, reflect the existing terminology used in the neuroanatomical literature, and then ideally, we want to be able to automatically replicate this parcellation in individual subjects based on multimodal areal fingerprints. And so the 1.0 version of the HCP's multimodal parcellation has all of these qualities. So how should we go about this? Well, most extant parcellations were generated using only one of the following modalities: architecture, function, connectivity, and topography, but the HCP measured all four. And we're going to use gradients or the derivative with respect to space of each of these measures as an objective measure of where the different modalities are changing rapidly. And these represent potential areal boundaries, and they also give us a sort of a common space in which to compare across different modalities. So what makes a gradient convincing as an areal boundary? Well, if we've got agreement in spatial location of that putative boundary between two or more independent modalities, that's really important. If we've got statistically robust and significant evidence of multimodal differences across the boundary, presence in both hemispheres, it's not associated with any known imaging artifact, we have prior information from the literature. And then the last thing we want to do is take this set of candidate boundaries and see if any of these areas have been found previously in the literature to confirm the existence of those areas or to describe any new areas that we find. So to cover the different kinds of information that were used in the parcellation, looking at myelin here, our myelin maps, the reds and oranges and yellows are high myelin, whereas the blues and purples and blacks are low myelin. And so we're going to focus on where myelin content changes across the cortical surface. And so the gradient is now going to have these reds and yellows and oranges at the locations of high change. And it's the local maximum of these gradients that's the most likely location of the areal borders. Now, some of these transitions are larger than others, but again, as I said earlier, it's the transitions that occur in multiple modalities that are especially interesting as areal border candidates. Another architectural measure we have is cortical thickness. So here we may not have quite as much information as a myelin map, but we still have very interesting and useful information. For example, the distinction between primary sensory cortex here and primary motor cortex has a really strong gradient between them. And here we've also regressed out the curvature because we're not interested in the fact that gyri tend to be thicker than sulci. That's just something that we know. We're interested in how different cortical areas have differing cortical thickness. We can use a very similar technique with task fMRI data. So here's listening to a story. Positive areas have more activity during the task, whereas the negative areas have more activity during rest. And we can take a gradient of this task fMRI beta map, just like we took the gradient of the Myelin map or the thickness map. Here we're using the effect size maps, and that's a more biologically interpreted measure. What about resting state functional connectivity? So here we have a seed, this white dot here, and the yellows and reds are said to be functionally connected, correlated, and are positively correlated. And we can look at the gradient and functional connectivity patterns across the cortex and step across these gradient boundaries here. We see the functional connectivity pattern changes as we step across these strong gradients. Two patterns that are relatively similar to each other, but in between a very different pattern here. And also, this particular pattern happens to look very much like our language story contrast. And it goes to show that areas that are functionally connected are often activating together. What about topography? Not enough time to discuss it in detail, but we can map out visuotopy with either retinotopic task or with resting state fMRI. In either case, these allow us to map visual areas where connectivity would tend to split areas along topographic gradients, such as grouping foveal V1 and V2 together and peripheral V1 and V2 together instead of giving us area V1 and area V2. We can also map out subareas of somatosensory and motor cortex. So let's put it all together for one cortical area to give you guys a flavor of how this process worked. So we're going to focus on this lightly myelinated cortex here in posterior frontal region. It's right anterior to the motor strip here. There are eye fields on either side of it. So we put these white dots along the gradients in the Myelin map, most likely areal boundaries. Then if we look at our task fMRI data listening to a story, we see that there are gradients in the task fMRI data that nicely match the gradients in the Myelin data. We can look at functional connectivity, putting a seed in this perisylvian language area here, and see that it has unique connectivity to this particular cortical area, whereas the seed in here, LIPV, this heavily myelinated hotspot, has connectivity to the two eye fields, but not this particular area. And it makes sense that we have strong connectivity gradients here, basically between this area and the eye fields on either side of it. So we have multiple independent modalities, architecture, function, connectivity, all agreeing on this particular area. And so the last step is to see if this area has been identified previously in the literature. And it turned out that Adolf Hopp in the 1950s had made these drawings of myelin content on the cortical surface. Here's the motor cortex and the two eye fields, and he found a more lightly myelinated area right in between them and called it 55B, which is why we've stuck with that name. All right, so we're going to construct this parcellation from the 210P group of subjects that David had mentioned. We're going to define the borders based on the group average data, that highly reproducible data that David was showing us. Architecture, Myelin maps and thickness maps. Function, the task fMRI maps. Connectivity from resting state connectivity. And topography, visuotopic resting state connectivity. And we're going to use a semi-automated approach. So David and I chose the modalities that were useful for each areal boundary and drew an initial boundary. Then we used an algorithm to optimize that boundary to make sure it did as well as one could do computationally at following the multimodal gradients. And then we identified the areas with respect to the prior anatomical literature, trying to keep the areal names the same when we could. So we went into this with a few predictions based on prior monkey parcellation that David had made and an extant at that time partial human parcellation. We thought there would be around 150 to 200 cortical areas per hemisphere. We expected wide variability in areal shape and size across the cortex. There would be examples of interareal heterogeneity, such as the sensory motor topographies. So what did we find? But we had 180 areas in each cortical hemisphere, and we were able to identify every cortical area in both hemispheres. 97 of those areas had not previously been described, and 83 were areas that have been previously found. And there's wide variability in the areal size and shape across the cortex, anywhere from the thin strips of sensory motor cortex to the horseshoe-shaped areas in visual cortex, and almost any shape in between. And we were able to parcellate, regions like, primary motor cortex M1 or primary somatosensory cortex, or the visual areas that have been troublesome to unimodal, fully automated algorithms before. But we do certainly note that some of these larger areas may contain subareas, like you could have a face subregion of sensory motor cortex containing multiple subareas for motor and primary sensory cortex. We colorize the parcellation according to the main input streams of the brain and two anti-correlated cognitive networks. So we have our auditory stream in red, sensory motor stream here in green, and visual stream in blue, and then task negative or default mode in the darker colors, and task positive or anti-default mode in the brighter colors here. And then any of these areas that have strong contributions from more than one modality, such as LIPV with strong somatosensory motor and visual contributions, are intermediate colors. So now that we have this map of brain areas, how can we use it? Well, here we have a Myelin map with the brain areas overlying it. And this Myelin map is around 60,000 data points, one value for each surface vertex. That's kind of a lot of data points. And if you wanted to do, statistical tests, that means you're going to do 60,000 non-independent statistical tests. You're going to have to do some sort of a correction to deal with all that. But, we can sort of short circuit that whole process by just averaging within these cortical areas. So the pattern you see in the parcellated Myelin map versus the dense Myelin map is very similar. But we've decreased the number of tests, the number of data points remarkably down to only 360 from 60,000. So that's a major benefit of parcellation. Parcellated analyses allow us to do functional connectivity analyses. Here we're looking at the default mode network and full correlation and partial correlation. That's a bit more of a sparse map here. Task Fmri is another place that benefits. So here we have a task Fmri contrast, working memory contrast here. Here's it is, all of the HCP's task contrasts in this matrix here. Here's the story contrast. And if we look at this story contrast and compare it to what we would have gotten with what we call the gray ordinate-wise analysis, we can see that our z-statistics are clearly higher in the parcellated analysis than this gray ordinate-wise analysis. Now, if they were the same, we would have all these blue dots right along this line of equivalence here. But you can see that, if we consider, all of the points, they're doing the averaging the time series within each area and fitting the GLM is a much more, gives us much more statistical power than fitting the GLM to every single gray ordinate time series and averaging the results that we get. So that's why parcellating early can be very helpful for task Fmri. So a quick case study on sharing extensively analyzed data. So this paper that described the parcellation had over 50 figures and over 200 data files. And so sharing that in a sensible way that was manageable for us to do and also well organized for people who were interested in the data was a challenge. So we have these scene files in the Connectome Workbench software, and we actually generated all of the manuscript figures that were in this paper. Within Connectome Workbench, these annotations and images and things that you see were all actually generated in Connectome Workbench, almost as if we were using PowerPoint to make the figures. And then we could save the scene with the state of Workbench and come back to it later and edit it if we needed to. But when it came time to share the data, we could upload those scenes directly to Balsa, and along would come the data files needed for each scene. We put a link in each figure of the paper to the data in Balsa, which you can click the link and see the figure in Balsa, and download all of the data needed to exactly reproduce what you were seeing in the paper. If you were interested, there's a a fully labeled version of the parcellation that's available on Balsa as well. So let's talk about reproducibility of this multimodal parcellation. There's several questions we should address. How to replicate the group average parcellation in individual subjects. We ideally want to do that automatically, given the enormous amount of work that it took to make the single group average parcellation. We want this to be relevant to atypical subjects. So, a group average is going to give us the most typical or most common pattern of areal arrangements in humans, but we want our parcellation to be relevant to those who don't have that arrangement of cortical areas. We want to be able to replicate this group average parcellation in a new group of subjects, and to address all of these issues, we developed a machine learning areal classifier. So in terms of reproducing the areal classifications at the individual level, we have scanned some of our HCP subjects twice and run them through the entire HCP protocol 2 times. So here we have a folding map, the group location of area 55B, Myelin map, we can see 55B slightly myelinated, a language connectivity map, we see that it's connected, as we saw earlier. The raw output of the areal classifier is what we see here, and the final output is here. So this isn't a hard case, because we use the areal feature-based registration that David mentioned, we have area 55B already aligned with the group average location. We are able to see it again using the areal classifier a second time. So this is an easy case. We don't actually necessarily need the areal classifier for this case, but what about something atypical? So here we've got this stripe of high myelin going right down the center of area 55B. There's a lack of connectivity in the language connectivity map here, and we see as a result there's a split in this area 55B in this particular subject in the raw and the final outputs. So that's what we see if we run the subject through scanning and analysis once. If we do it a second time, we see that this difference in cortical areal layout is highly reproducible. They basically have the same split, look very similar to each other. Here's a different pattern. We've got high myelin right at the bottom of the expected location of 55B. The language connectivity is shifted upwards. And so we see a shift in area 55B in the raw and final outputs, and that shift is reproduced in a test-retest setting. So this areal classifier is reproducibly able to identify area 55B, and these atypical areal topologies are not some artifact or noise. They're real. They're stable across time and highly reproducible. So what about group parcellation? So here's our group parcellation. Here are some individual parcellations. We can see that that while there are strong similarities to the group, each individual has significant differences as well. So how well is the areal classifier able to find these cortical areas? Well, it was trained on the parcellation data set, the 210P data set. So we would expect it to do well, and it does. Basically, it finds almost every area and almost every subject. It's all yellow here. This validation data set, the machine learning algorithm never had a chance to see this. So this is its true performance. And we get 96.6% of all cortical areas in all subjects identified in the validation data set. If we drop the task Fmri battery, we do almost as well here. And so this means this areal classifier will work for studies without the HCP's specialized task Fmri battery. We need the T1, T2 field map and Fmri to use it. So we can take these individual subject classifications and make probabilistic maps in the 210P and 210B data sets. Here we can see some examples. So some of these probabilistic maps like B1 or M1 are very tightly localized, but some are a little bit more spread out, like 55B, where we're seeing some of those atypical patterns in different subjects. But these probabilistic maps are highly reproducible across these two independent groups. What about reproducing the overall parcellation, a maximum probability map? Well, we can compute those from the probabilistic maps here. We're displaying the areal boundary gray ordinates. Anything that's blue is a boundary for 210P. Anything that's red is a boundary for 210V. And then purple means they overlap, and you can easily see that this is mostly purple map. The boundaries are in very high agreement. The correlation of the two parcellations in 210P and 210V are very high. And this is importantly in line with those high reproducibilities of the original data that we mentioned earlier. So that's an important finding. But what happens if you try to compare traditionally processed results to the new map? So here's our original map here. This is work that Tim Coulson did with us. And we're going to process these individual areal definitions. We have, 449 of these that we actually made for individual subjects, and we can take them. They exist on subjects' native surfaces, but we can put them in the volume. We can resample them through different surface registration algorithms, do whatever we want to them to simulate what happens when you process your data in different ways. So if you use the recommended HCP approach with aerial feature-based registration and without smoothing, you are able to reproduce the map as one would expect. And the uncertainty in the locations of the cortical areas is really only at the border where you would expect it to be. In the centers of areas, you don't have much uncertainty. But if you use traditional approaches, such as volume-based alignment with and without volume-based smoothing, you just have markedly higher uncertainty. Extends beyond the boundaries. There are significant swaths of cortex where we're in yellow, meaning that we don't even have highest probability gray matter. There's CSF or white matter is higher. We can, this kind of stripey here, so we can do the surface volume to surface mapping a different way that gets rid of the biases, but it actually increases the uncertainty even more. So the point of all this is only the HCP's approach is able to reproduce the map of most cortical areas. So that's our studies, but what is happening in other people's hands? So here are HCP retinotopic data, and here are retinotopic data from another group who processed their retinotopic data, as David mentioned, like almost everybody else does on the cortical surface. And despite the fact that they didn't use the aerial feature-based registration, they used the folding registration, we can see that these retinotopic maps are really quite reproducible. The HCPs are a bit sharper because of the aerial feature-based registration. Population receptive fields are a little bit tighter, again, because we've got higher alignment. But overall, these are very similar patterns. What about cognitive neuroscience? Well, Mozart's Asim and John Duncan in the UK used the HCP's data to define what they call the domain general cognitive core. So these are young adult HCP data that they used to do this. Here's their cognitive core here. And so this is their overall pattern. And they wanted to see, could they reproduce this pattern in their own subjects that they'd scanned locally. So they scanned their subjects with an HCP-style protocol, analyzed the data with the HCP pipelines, and lo and behold, they get basically the same answer as they got from HCP data. So we really have high reproducibility, even of cognitive neuroscientific findings when using HCP-style approach. So I'm going to close where we began. Just a reminder of the HCP-style approach to brain imaging research. These principles, again, acquiring high quality, high resolution multimodal data, avoiding blurring brain imaging data or lossy temporal denoising, using 2D surfaces for the cortex and 3D volumes for subcortical nuclei, accurately aligning brains across people, using an accurate, functionally relevant map of the brain, freely sharing brain imaging results in publicly available databases, not just peak 3D coordinates. And just to really hammer home this point, that if you use the traditional approach with volume-based alignment, volume smoothing on those individual subject cortical areas, what you get out of it is something that looks like this, which is really quite blurry, and no particular voxel in this MNI space has a particularly large amount of area MT in it. Whereas if you do the alignment and don't use the smoothing in the surface space, you can get very high overlap. And it's analogous to the difference between ground-based and space-based telescopes before the advent of adaptive optics, where you just got much sharper images going to space than you did at the ground. And the benefit here, as David mentioned, is you don't have to spend billions of dollars on a space telescope. You just have to process your data differently. The benefits are highest if you get high-resolution data, but as David mentioned, even legacy data will benefit. All right, I think that's all we have. Happy to take any questions, as I'm sure David is as well.
Fabulous. Thank you very much. That was very illuminating. I will invite the audience to either intrepidly attempt to unmute themselves and ask a question, or use the chat feature, and we will try to negotiate. If you want to maybe stop the share, we may see some of our friendly faces. And also, I realize we're coming up sort of on the 3 o'clock, and people will probably be leaving. And also, thank you very much. And I see some clapping there, which I'll do in person, I guess, if I turn my video on. Thank you. So I guess while we wait for other questions from, if there are other questions from the world, can you remind me? So you showed the individual parcellation, techniques, and yet you have, sort of the standard ones you make available from your, 200 and, however many subjects. Is it recommended that for each of my studies that I'm doing, I should go off and do my own individual parcellations, or am I really okay just using some of your parcellation templates, that are made available for my regional types of analyses?
Well, at this point, we've done some stuff with the individual parcellations. Most of what we've done also has been with the regional approach. So if you do the aerial feature-based registration, most subjects you're going to have, you're going to get lined up pretty well. But, as I showed with Area 55B, there are exceptions to that. And so in the long run, once we've made available this pipeline, it's been written up, and we're actually, we're doing some stuff to get the temporal ICA stuff out first, and then the next thing that'll come out will be the aerial classifier. And, it'll depend on what you're interested in doing for your particular study and how many pipelines you want to run. One thing you do have to think about for the individual subject aerial classifier, and something that we also need to think more about, is what happens if you don't find a cortical area. So we said, 96.6% of areas were found, and that sounds pretty good, but it means, 3.4% weren't found. Are those, is that just because the data weren't very good, or there was some issue there, or is that real difference? Are there missing areas in some people? Do some people have extra areas? And that's something that we plan to investigate further. We're funded to investigate that, but it's something we don't know the answer to yet. So this is, we're still sort of moving forward in this. And so I'm giving you an answer is probably we'll want to use the individual ones in the end, but we're not quite there to make, the explicit recommendations. I do see JB Pauline has.
Oh, hi. Congrats, David and Matt. I think this body of work has really been completely transformative of the neuroimaging fields in general. To me, it's like the equivalent of the early MNI template, sort of like a trailblazing work. So I think it's fantastic. So the thing that struck me, because I did work a bit on parcellation myself, with my former postdoc, Bertrand Tillon, and others. The thing that struck me, you're proposing one parcellation that is fit, it somehow is, and when we're doing some machine learning and trying to predict, for instance, we realize that the number of parcels that will actually be the best fit for a prediction model is actually not always. It will depend on the task. We depend on so we find sometimes the 1,000 parcel is actually much better predictor, which will make a better set of features than 210 parcel, or than the. So I'm just trying to find what's your take on this aspect of how variable the actual, the fitting aspects of the parcel can be depending on the task, and what is your take on should we propose you're proposing one kind of generic parcellation? What's your take on this variable task aspect that could lead to other at least number of parcels, and it could be sub parcels, of course.
Well, so David will probably have something to add to this, but I sort of think of it in terms of the hierarchy of brain organization. That you probably heard the idea that we start with the highest level hierarchy is the functional system, and then below the functional system we have the cortical area, and then below the cortical area we have cortical columns and things like that, or some of the sub-regional organization that we see in sensory and motor cortex. So what we were trying to target here was the cortical area level. We were actually trying to hit one of those particular levels specifically. This is version 1.0. It would be more surprising to me that we don't have things that can be improved upon it than that's the perfect answer. That's unlikely. There's I expect that there will be a version 2.0 at some point. But this idea of multiple levels, I've always tried to think of it as targeting a particular level of the brain organization rather than just sort of doing a sliding scale of numbers of parcels. There are many automated algorithms that can do that. You say, I want so many parcels, and that's how many it gives you. There are others where the algorithm attempts to estimate the number of parcels, and you might have some cost function, and you find that there's a minimum here, and there's another minimum here, and so on. That's, we can think about dimensionality reduction. Going like that. Certainly, ICA can have different dimensionalities. There's this idea of wanting to have functionally homogeneous parcels that's out there. And to me, if that's your goal, if functional homogeneous parcels is your goal, why not use a weighted spatially overlapping parcellation like ICA, where you're going to get by using the multiple linear regression, you're going to get signals that are pure, even if they're spatially overlapping, because that's what the multiple regression is going to give you. So, David, what do you want to add?
I would add to that an informative example, which is the fusiform face complex that we identified in our map. And we called it a complex, not an area, because we were aware of wonderful study by Nancy Conwisher and Marge Livingston and others who've identified face patches in humans and others in monkeys, particularly as Doris Chow and colleagues have identified. So if one looks at the HCP data within the fusiform face complex, we find in some individuals there are distinct face patches that tend to lie within FFC, but they're quite different from one individual to another in their number, their distribution, and even between one hemisphere and another in each individual. So that's an illustration of a finer granularity that is challenging to represent at a group level, or when one represents it at a group level, one loses an interesting aspect of individual variability that our particular MSM aerial feature doesn't align perfectly, and it's quite possible that the topology is between different individuals is such that you can't align them perfectly. So, and also in that inferotemporal cortex in monkeys, Doris Chow has shown a complex pattern that has some commonalities across individuals and again some quite striking individual differences. So it's a work in progress, both in terms of analyzing the data and getting the proper conceptual framework for thinking about areas, sub-areas, patches, and so on. So I think it's an intriguing area for continued discussion and exploration.
Thank you so much.
I think it's sort of, building on that a little bit, maybe every normal human has the same functional systems, but maybe, we can already see cortical areas that there are these topological differences and that need more exploration. But as you get to the patches, there's more likely to be more topological, differences, the columns, your ability to use something like, aerial feature-based registration, even a topology preserving registration, you'll run out of being able to do that at some point. And at some point, you're going to have to be doing things like the aerial classifier or stuff like the hyper alignment that has been done. And almost certainly we can't align our brains neuron to neuron. So where we might start with agreement in functional systems, that will gradually break down as we get to finer and finer scales.
Great, thank you. Any more burning questions? I know we could usurp your entire afternoon with lots of questions, and some of those will have to wait till next we see in person or virtually. But I wanted to give the audience another moment if there was a question. Seeing none, I will go back to thanking you again for joining us, and thanking you for being one of our collaborative projects for the ReproNim project. And we will keep working on ways to integrate some of the ReproNim philosophies and technologies with the tools and procedures that you all are promoting. So thank you for your past and future participation with us. For those on listening, next month's webinar will be Friday, November 5th. It'll be Chris Riordan. I'm not exactly sure of the topic, but you know him from DCM to NII conversion and Micron and all sorts of cool tools. And we'll see what he's using those for in application areas. So thank you all, and see you next month.